Hello. In this lecture, we're going to be reviewing microbial metabolism. Now, this is not just run-of-the-mill, okay, let's talk about aerobic and anaerobic metabolism, glycolysis, Krebs cycle, electron transport chain. Actually, we're going to show you how a lot of this plays an important role as a background to understand some of the microbial tests that are actually done to determine the identity of certain organisms. That'll be the latter part of this. So what we're going to do is move forward now. When I talk about microbial metabolism, we have sort of a catabolic and anabolic reactions that are the yin and yang of metabolism. Keep in mind that metabolism is really the collective of chemical reactions that break down products for energy or build up products to more complex products. That's why we have both sides of the coin, catabolism, anabolism. Catabolism pathways, these are chemical reactions to break the substance down to obtain energy. These reactions tend to be hydrolytic and exergonic, meaning releasing energy as they are occurring. Anabolic pathways are chemical reactions which make use of energy and simpler compounds to make more complex products. They tend to be endergonic, in other words, requiring energy to occur, and use dehydration synthesis as part of the chemical reactions. Now, when you have an amphibolic pathway, this is what we refer to as a dual or reversible pathway. That can be either anabolic or catabolic. If you take a look at a lot of reactions, biochemical pathways consist of reactants, intermediate products, then end products. The IP, intermediate products, may be fleeting, or they may be something that you can actually extract and use. But we're going to look at all of this as we move forward. Here's a nice diagram that kind of summarizes it. Catabolic reactions, we basically are breaking complex molecules down to simpler ones and obtaining energy in the process. Anabolic reactions, though, are taking simpler molecules, require energy, and you end up with more complex molecules. Simple example. If I take starch, I break it down to the individual glucose molecules, and then I break those down further, I'm going to get energy out of this. Turn it around. I take glucose molecules with some energy, and I'm going to make them into longer, more complex molecules, such as starch or cellulose, etc. Now, one of the key points you want to keep in mind is ATP, adenosine triphosphate. It's like metabolic money, which sounds kind of goofy, but really what it is that, remember that chemical energy in a cell primarily is ATP. There are other triphosphate nucleotides. Uridine triphosphate, guanosine triphosphate. ATP is required for a cell to do work. Some of these other unique processes have been found where certain chemical reactions occur and require UTP or GTP. Now, cells that go through a process of breaking the phosphate away from ATP and recharging ADP to ATP by adding a phosphate. This is sort of like this ATP-ADP cycling. Some cells can strip away one phosphate from the ADP and get AMP, adenosine monophosphate. So you start off with 3 phosphates, you go to 2, and in some cases you can actually strip it right down to 1. Every time you break the bond with the phosphate, you get a certain amount of energy. Here we have the ATP here, 1, 2, 3 phosphates. It will release energy for anabolism as I break off one of the phosphates. I have ADP. Now, What if I want to recharge ADP to become ATP again? That uses energy for the catabolism. And it basically means that you're going to have some energy source to add on that phosphate to give it back to ATP. Here is a nice way of looking at this entire molecule. Here you have the nitrogenous base, you have the sugar, and in this case it's ribose, and you also have 3 phosphates attached. And you can see that you'd have adenosine monophosphate, diphosphate, or triphosphate, depending on how many phosphates are attached. Cells rely on enzymes for metabolism. Basically, a lot of these chemical reactions, to maintain and have them occur at relatively modest temperatures. When I mean modest, body temperature, 25°C, 30°C, something else like that. When in nature you might not get that unless you had enzymes present. It's because of the enzymes that you can have these chemical reactions occur under the normal cellular conditions. So enzymes are protein catalysts. Catalyst is a substance that speeds up specific chemical reactions. Reaction rate, this is the rate of chemical reactions over time. Most of the enzymes usually use the name of the substrate plus ASE for the suffix. And what's the substrate? Well, that's what the enzyme acts on. Okay? So here is a brief review table that I encourage you to take time to review. General enzyme characteristics. Enzymes are biological catalysts. They're effective in small amounts. They act on specific substrates to generate specific products. They increase the rate of reaction. They provide a route for reactants to products that has a lower activation energy. Remember, it's activation energy that plays a critical role. You could get certain chemical reactions if you had a much higher temperature, but then again, you wouldn't have it being at that temperature sustained in a living cell. With enzymes, you can have this in a living cell at what would be basically what you might call regular temperature, regular conditions. Now, enzymes can be regulated to control biochemical pathways, and some enzymes require cofactors to be active. Keep in mind, as with any catalyst, when you are having a chemical reaction occur, the catalyst is not consumed nor permanently altered in the reaction. Okay? Enzymes are genetically determined. There are specific genes that carry the code necessary for the proper structure and hence the proper function. Okay, so we've got all of that. Let's move forward. Now, enzymes are classified based on the type of chemical reaction catalyzed. There are a variety here. I'm not going to have you memorize all of them. Don't sweat that, but just to get some idea. If I say it's an orthoreductase, it does basically oxidation reduction. If it's a transferase, it will transfer one functional group from one molecule to another. That functional group might be a methyl group, an alcohol group, or a hydroxyl group. It might be a particular amine group. When we talk about a hydrolase, these have hydrolysis, the addition of water to help break the bonds. So hydrolase, for example, sucrase, let's just take that one, breaks down sucrose. And so you now end up with both fructose and glucose as what were the former building blocks to make a molecule of sucrase. Lysase, this is removal of groups of atoms without hydrolysis. Isomerase, the rearrangement of atoms within a molecule. Now, if isomerase doesn't sound familiar, there's one that you know. You probably had some Coca-Cola or some other cola product recently. Now, not the diet stuff, the regular stuff. And what they did is they used a particular isomerase called invertase. You see, very rarely do they use glucose anymore or sucrose to sweeten up your soda. what they do is they get corn syrup, and they use invertase that basically will flip the molecule around in such a way so that it comes out like fructose. That's where you get what's called high fructose corn syrup. And the neat thing is, obviously, it's going to be somewhat a little bit sweeter, and it's going to be a lot cheaper in production, and everybody benefits. Now, ligase is joining of two molecules, usually using energy. You've heard of DNA ligase. We've talked about that before. Okay, let's move forward. Now, one of the things you have to think about is this. You have the enzyme and you have the substrate. The substrate is the molecule that's going to be acted upon, right? So scientists were trying to figure out how did these things work. And first they came up with the lock and key model. That was the original theory. The substrate's the key, the enzyme active site, and there's a particular site on the enzyme where the substrate fits into it, okay? Kind of like a key and a lock. The active site is where the substrate not only binds down, but has the chemical reactions occur. Sounded nice, but there were certain things that didn't seem to fit until further observations occurred. And then we moved up to the induced fit model. This is the current theory of enzyme-substrate interaction. The enzyme molds, or actually flexes, around the substrate. Now remember, enzymes are proteins, so they can have some flexibility in them. So when the substrate fits into the active site, it's almost as if, think of it sort of like the enzyme flexing around or oozing around that particular substrate to get a better hold. And so it produces an induced fit. And that's when you have the chemical reaction occur. Now, enzyme-substrate complex is when you have the enzyme and the substrate come together. Okay, that sounds great. And as a result, you have a much lower activation energy for that same chemical reaction. Because remember that energy is required for the initiation of any chemical reaction. Enzymes reduce that activation energy for the reaction to occur. And if you see in some of the charts, you've probably seen this before. The differences between chemical reactions, same chemical reaction, with and without enzymes. Now, by the way, there is a particular state I want you to keep in mind, and that is the transition state. It's the point where substrates are becoming product, but they're not quite complete. It's kind of like an intermediate form. So what you see here, right off the bat, here's the enzyme substrate induced fit model. Now, you have the substrate, the active site, the enzyme. Substrate fits. Now what happens is you notice it kind of like some parts of the enzyme kind of flow around or pucker around or get right around that substrate. And now you're having a good hold, chemical reactions occur, and in this case you're breaking the bond between these two parts of the substrate, and they become products. Once they become products, they pop out of the active site. Okay? Now, by the way, you notice it says, and the enzyme is again available. One biggie here. This reaction can occur hundreds, thousands, or even millions of times per second. Okay? And since the enzyme is not chemically altered in any way, shape, or form, it can keep doing this as long as there is substrate present. Okay? Now, here is the famous chart that everybody sees, and this is basically time, energy, either released or invested. Here we have our reactant. Without the enzyme, you're going to need a much higher amount. From here to here, you're going to need a much greater amount of energy that has to be invested. That's the activation energy, so that you can eventually get your products, okay? Started as a reactant, ends up as products. You had a certain amount of energy invested. Without that enzyme, you have a certain amount that's released as a part of the chemical reaction. Okay, that sounds nice. But here, follow the green line. if you react it. You have a much, much lower amount of energy that has to be invested. And this is because the enzyme is doing a lot of the work, and you still get the same amount of energy released. You get the same products. And by the way, notice the time difference between here and between here. So this can occur, this reaction, much quicker, as well as much less expenditure of energy is required. Now, enzymes require cofactors in some cases. Cofactors are the components that assist an enzyme to become a functional enzyme. They may include metal ions, organic molecules, etc. For example, an apoenzyme is an inactive enzyme lacking a cofactor. When it gets the cofactor, you have what's called a holoenzyme, basically functional enzyme, and it includes any of the necessary cofactors. Now, when you have cofactors that are metal ions, many of them exist: iron, zinc, magnesium, calcium ions. For example, the enzyme that we have in our liver called alcohol dehydrogenase, yes, it's involved in the breakdown of alcohol, requires zinc. Now, there are other types of helpers or cofactors. They're classified as coenzymes. These are organic cofactors for enzymes. So they contain some form of carbon. They may be a very simple molecule or very complex. For example, many coenzymes include vitamins or molecules made from vitamins. And we'll get into that in a chart you'll see in a minute. Now, coenzymes NAD plus and FAD, I believe that's FAD plus, excuse me. These are electron carriers. They shuttle electrons from one reaction to another, okay? Now, what happens here, oh, and I stand corrected, it's supposed to be NADP, I believe. But anyways, what happens here is that these are actually derivatives of niacin. Nicotinamide adenine dinucleotide, which is NAD. Nicotinamide mononucleotide phosphate, NADP, are electron carriers made from niacin. Flavin mononucleotide, FMN, and flavin adenine dinucleotide, FAD, are electron carriers, and they're made actually from another B vitamin, riboflavin. Now, coenzyme A is originally derived from pantothenic acid, which is actually called vitamin B5. Yeah, they're all from originally vitamins, but they are finding that coenzyme A is actually essential in the Krebs cycle reactions, which we'll go into in a little bit. Now, just to give you a heads up also, there's a unique group of RNA molecules. They're called ribozymes. These are RNA molecules with catalytic properties against other RNA molecules. So here's my cofactor, here's my enzyme. And like this fits in sort of like a shoehorn here, or right underneath here, so that basically the substrate attaches, fits in nicely with the active site, products are made. Without that cofactor present, fitting in right there, the substrate may bind, but you're not going to get products made. Now, before you sit there and look at that list and go, I don't want you to memorize all of these. It would be helpful if you go over them. Because interestingly enough, many of the B vitamin coenzymes started off as B vitamins, yes. And by deficiency, that's where you see a lot of different illnesses show up in humans, whether it's anemia, whether it's pellagra, whether it's dermatitis or spinal bifida or pernicious anemia and nerve degeneration. But you see, here's part of the point. They play a key role in the function of so many other enzymes because they are coenzymes. They help and assist in that function. Okay? Now, there are many factors that affect enzyme activity. Because it's protein-based, temperature will be one of them. Lower temperature can slow chemical reactions. Yeah, we know that. Higher temperatures can speed up chemical reactions, but there are limits because of the fact that enzymes are protein-based. And so you're going to have an optimal temperature for the organism and for the chemical reaction. You go beyond the optimal temperature, too high, and the presence of that amount of temperature will denature the enzyme by denaturing its protein structure. Loss of structure, loss of function. Very straightforward. It's interesting to note, though, that there are some microbes that are thermophiles. And this is where sometimes it gets interesting. They have unique proteins that function at very high temperature and have useful industrial and commercial properties. For example, Thermus aquaticus. That's basically where we get the DNA polymerase that's used in the process of the technique called polymerase chain reaction. Now, this is what I was talking about. It's a nice chart here. Reaction rate, so that would be so many reactions occurring, and this would be the temperature. You notice that you start off low and slow here. At low temperatures, you're going to have just a little bit of the reactions occurring. But how come we don't have this curve continue to go off infinitum? Well, if it was an inorganic reaction, we don't need any enzymes, etc. It might continue to spike upward. But what you notice is the optimum temperature here, right around the 37 degrees or 40 degrees Celsius. What happens? Then you start going and crashing downward. The reason that's occurring is because the protein of the enzyme is denaturing. It's losing its secondary, tertiary, and even in some cases quaternary structure, and therefore its function degrades quickly. What we also have are other factors that can influence the function of enzymes. pH. Above or below the optimal pH will alter enzyme structure. That will denature the enzyme, and it leads to reduced reaction rates. Some organisms, though, you have to be aware of, have that capability to adapt to unique pH ranges. For example, Helicobacter pylori, the organism that can get into the stomach, lead to stomach ulcers. It can withstand very, very low pH, which you would expect to find in the stomach because of the secretion of hydrochloric acid into the stomach lumen. They are able to avoid the complete destruction of the cell and still proliferate. Here is an active protein. Here is an inactive protein. Why is it inactive? Look at all of that. Alpha helices, beta pleated sheets. Well, now you don't. You just have this big long chain, which would be basically the primary sequence and the primary structure of the protein. Now, how do we regulate enzyme activity? Well, cells can regulate enzyme activity or the amount of enzyme present or adjusting enzyme activity via several of these processes. Let me give you one pointer here. Nature abhors wasting resources. What? It hates to see production of proteins that it does not use. It does not enhance the survival of an organism to make stuff that you're not going to use. That's the basic gist here. So I either shut off the enzyme when I don't need it, or I destroy it. But I certainly don't keep making stuff that I'm not going to use. Let's talk about this. There are different means to stop or inhibit the activity. First is phosphorylation state. If I alter the activity of an enzyme by adding or removal of a phosphate group to the enzyme, that is how I can basically keep enzymes either active or inactive. Kinases are enzymes that add phosphates. Phosphatases are enzymes that remove phosphate groups. Now, another way to do this is inhibitors. These are compounds that block, and I mean block by inhibit, enzyme activity by attaching to the enzyme. There are several different types of inhibitors. We'll go through one of them first, the competitive inhibitor. And by the way, these inhibitors, some of them you've probably taken. What do you mean by that? Well, some are antibiotics. They inhibit the action of a particular enzyme within the ribosome or in certain other places where enzymes are necessary for the survival, the function, or the structure of that bacteria. But let's keep moving and talk about competitive inhibitors further. As I said, they're compounds that compete. They're basically trying to vie for the active site of the enzyme. Therefore, the more that they're attached, that reduces or blocks the enzyme activity. An example of this is sulfanilamide versus para-aminobenzoic acid, PABA. Now, PABA is necessary for folic acid synthesis, which folic acids are also necessary for eventually DNA synthesis inside of a cell. But sulfanilamide is interesting because it is an antibiotic. It binds to a particular enzyme in the folic acid synthesis pathway. As a result, you get a plummeting amount of PABA made, a plummeting amount of folic acid made, and therefore you can shut down DNA synthesis, perhaps even kill the cell. Now, you want to keep in mind something though. Excessive substrate can override competitive inhibitors that are present. What I mean by that is if you have a whole whopping amount of substrate, basically just by sheer numbers, they can more often get in, bind to the active site versus the inhibitor. Okay. Here is an example, substrate concentration and product formation rate. Maximum reaction rate with lower enzyme concentration, the lower the enzyme, the lower the number of, or the smaller the number of active sites, and so you're going to get this saturation here. If you up the amount of enzyme present, okay, you can go much higher in the maximum reaction rate because you have more of these active sites present due to the presence greater number of enzyme molecules present. Here's what I was talking about before about PABA versus sulfanilamide. If you look at the overall molecule, very similar. The only key difference is at this our carboxylic acid site. Instead of carbon, you have sulfur, and then off of the sulfur you have an amide group, and now you have double ketone structure. No problem. All of this is still worthy of being used as an inhibitor. Now, if I have more substrate than basically the inhibitor, I'm going to have a non-inhibited enzyme, and you can see why here. If the deep purple is the reversible competitive inhibitor, reversible means it attaches for a while, pops off, attaches for a while, pops off. What you've got is that with more and more substrate, you can basically swamp or overwhelm the actions of the small amount of molecules of the inhibitor. But flip it around. Remember, enzyme active site, if I had more inhibitor, I would be able to swamp out and thereby keep inhibitor in the active site more often than any of the substrates, and thereby break down the enzymatic chain of synthesis, for example. Let's go to the other one, non-competitive inhibitor. Now, these are compounds that decrease the enzyme activity. Keep in mind that we've talked about the induced fit model. The proteins flex a little, right? Okay, in this case, what we have is a binding site on the enzyme other than the active site. There's a lot of these sites And if you have the right molecule, it'll bind to this other site, but cause a twisting, a warping, a distortion of the shape of the entire enzyme protein, particularly at the active site. So, loss of active site structure means loss of active site function. Now, this is where you can get allosteric regulatory factors, which are basically the site of the compound's binding on the enzyme is called the allosteric site. Allosteric activation is when you have the binding of all the allosteric site is activating the enzyme activity. But what you have in the other side of the coin is allosteric inhibition. Basically, allosteric activation, you have something binding to that allosteric site, it increases enzyme activity. Allosteric inhibition, binding to the allosteric site reduces the enzyme activity. Therefore, the rates of product synthesis depend on the activity of the enzyme and the amount of substrate present. Here's my non-competitive inhibitor. Here's my enzyme. Here's the active site. I pop this guy right in here, and what happens? Well, the substrate may even bind, but you're not going to get products made. Now, in this case, I have no inhibitor present, so I've got a nice, clean, uninterrupted structural integrity of the active site, and I have the substrate bind, and you have the chemical reaction that leads to the products made. With allosteric inhibition, yes, you can basically swamp out and basically shut down all all enzyme activity with sufficient allosteric activator. Even if a molecule binds to the active site, you're not going to have any enzyme activity, and you're not going to have any product made. All right? Now, finally, we have one last one in which we can change some of the enzymes, and that's feedback inhibition. This is a reversible process where the products could be whatever's down road in this feedback inhibition, and basically this enzymatic chain or enzymatic pathway, whatever the intermediates or the end product can modulate or turn off the activity of the enzyme earlier in the pathway. Usually it is the end product that acts in earlier steps of the pathway. So if you look at this, you have your pathway and the end products build up. We don't want to have too much of a buildup. So we modulate the entire enzyme pathway, tweak it downward. And as a result, we get less end product, and this may act almost in a homeostatic manner. Too much, shut down activity. Too little, turn back up activity. Now from here, we're going to talk about obtaining and using energy. Redox reactions, if you remember, those are reduction and oxidation. They're basically going to fuel the recharging of ADP to ATP. Now, reduction reactions are when you have an atom or molecule that loses electrons. Oxidation reactions are when an atom or molecule gains electrons. Redox process is linked. So one compound might lose electrons while the other one gains those same electrons. Many biochemical reactions, usually the oxygen is the common oxidizing agent and the hydrogen is the common reducing agent. Okay? So when I say something's a reducing agent, it's going to probably end up losing electrons. The oxidizing agent is going to gain electrons. Now, cells harvest energy from nutrients by oxidizing them, stripping electrons from them. Molecules with many carbon to hydrogen bonds, such as what you see with proteins, carbohydrates, lipids. They're highly reduced and readily oxidized to release energy. The more completely cells can oxidize nutrient molecules, the more energy they can harvest from these nutrient sources. Here's the main point to get you thinking. You compare the amount of ATP that is produced by just glycolysis alone versus aerobic respiration. You'll see this in a little bit. It's quite stunning because glycolysis, which occurs in the cell cytoplasm, can only get a small amount of ATP molecules for each molecule of glucose, whereas aerobic respiration, which not only includes glycolysis, but it encompasses the chemical reactions that will occur in the mitochondria. That's Krebs cycle, the electron transport chain, get a much greater number of ATP molecules as a result per molecule of glucose. Here we see this, we have oxidation. So we have a reducing agent and it loses an electron. We have an oxidizing agent and it gains an electron. NADH. Notice it has a hydrogen group. As such, we have it reduced. And when it's reduced, it has that hydrogen there. Okay, so reduction is a gain of hydrogen and 2 electrons. See, here are the electrons right here. When I want to oxidize it, though, I'm going to have NAD+. Why? I'm going to lose 2 electrons and lose a hydrogen. I've lost 1 hydrogen here, and I've lost 1 of the electrons here. And as a result, I have a plus charge here. Everybody clear on that? If not, go over it a bit. Feel free to give me an email of a question, okay? Redox reactions fuel the recharging of ADP to ATP. Coenzymes and redox reactions, there are 3 general mechanisms that make recharging ADP to ATP possible. Substrate-level phosphorylation, oxidative phosphorylation, and photophosphorylation. Substrate-level phosphorylation, the enzyme transfers phosphate from the donor to ADP, and hence, instead of adenosine diphosphate, with that added phosphate, you're ending up now having adenosine triphosphate. On top of that, you have oxidative phosphorylation. This is where redox reactions strip electrons from food sources, deliver the electrons to the electron transport chain. This has also been called the respiratory chain. This will drive ADP to ATP. And we're going to see that process occur. Finally, photophosphorylation. You already think photo, light. Well, light energy is used to activate the electrons and thus deliver electrons to the electron transport chain to drive ADP to ATP. Of course, you only see this really in photosynthesis. Now, this chart, although I do not say that you have to memorize it, this is a nice compact table here to give you a good review of the differences between the different 3 phosphorylation mechanisms, which recharge ADP to ATP. I encourage you to take time to review. Now, the catabolic process of cellular respiration, catabolic process. Carbohydrate catabolism, this is the breakdown of carbohydrates to obtain energy. Okay. Cellular respiration is a collection of redox reactions that use food to extract energy. The process includes glycolysis, an intermediate step, Krebs cycle, and the electron transport chain. Glycolysis, it's also called the following, which is a mouthful, the EMBDEN-MEYERHOF-PARNAS pathway, which is probably why to this day they all just say glycolysis. But basically, what does glycolysis mean? Splitting sugar. What sugar are we going to talk about? Glucose. This is really the first stage for aerobic and anaerobic carbohydrate metabolism. It doesn't require oxygen. We'll go over it in the chart shortly. After we've gone through glycolysis, we have an intermediate step. This is where we now have 3-carbon molecules, which are pyruvic acid. These are made into acetyl-CoA or fermentation products, which fermentation we will talk about later. Here we go. Now, what you can see in this chart is basically the outline we're going to face. Cellular respiration, otherwise known as aerobic or anaerobic. Now, what's the difference? Anaerobic is without oxygen. We can still have some cellular breakdown. We might have to use other means to get it, but even if it was just glycolysis alone, you're only going to get a couple of ATP out of it. From the intermediate step downward, this is all that you're going to see really in the mitochondria. You have an intermediate step, you have Krebs cycle, which is going to also set up the differences and the supply of electrons and hydrogen ions, protons, for the electron transport chain, which will give you the production of ATP. And with fermentation, though, the electron transport chain is either absent or the conditions do not allow it to operate. So what you get here is glucose going through glycolysis and then shunting off to a fermentation process. Now, you will get some ATP, but the fermentation products tend to be things that we would be interested in. Alcohol, acids, other types of synthetic products, or I should not say synthetic, but basically synthesized through fermentation. The fermentation will regenerate the NAD+, so that glycolysis can continue and some ATP is made. So, what are we talking about here? Well, with the prokaryotic cell, we have to know where certain events occur. If you remember the eukaryotic cell, hey, glycolysis real easy, all in the cytoplasm. Intermediate step in basically the matrix of the mitochondria. Krebs cycle, matrix of the mitochondria. Electron transport chain, inner membrane of the mitochondria. But what about on the prokaryotic side? Okay, we've dealt with eukaryotics. In prokaryotics, you're going to note in the cytoplasm you have glycolysis. You will also have the intermediate step. You will also have the Krebs cycle present. And it is only in the plasma membrane that you're going to find the process for the electron transport chain. Now you see these tabs up here, they're going to help you to review this. And by the way, before I have students that lose sleep, don't. You do not have to memorize each step. Okay? You have to get some basic summary of this, and that's what I'm going to have in the PowerPoint notes. But every one of these steps does a biochemical conversion and, of course, would have certain enzymes playing a role. Just keep that in mind. But you don't have to memorize all of the 10 steps, etc., etc. You start with a 6-carbon molecule glucose. You break it in half. Each part here is going to have some phosphorylation that eventually part of the reason it gets that is the supply of phosphate that will take ADP and convert it to ATP. But just like in industry, you've heard people say, well, to make money, you gotta have money. Okay, so you have to make some investment in the very beginning of glycolysis, and that investment is 2 ATP molecules. In the end, you end up making 4, and if you subtract the initial 2 that you had to invest, you end up with what is referred to as a net production of 2 ATP molecules per molecule of glucose. Notice that the molecule of glucose has been now broken down into 2 3-carbon molecules, pyruvic acid. You will also get a little bit of NADH produced, which will be used later on. You will also get the production of H2O. Now, the intermediate step, this is where it gets interesting. Remember, we started with 2 molecules of pyruvic acid. Each pyruvic acid is 3 carbons. You're going to have this little process, and you're going to basically also have some NADH made. And what you're going to do is have the coenzymes of coenzyme-A that is going to pick up from each of these molecules 2 carbons. Each carbon is going to, or I should say, each of these 2 molecules of pyruvic acid are going to lose 1 carbon, and that's going to basically make up carbon dioxide. And so the CoAs, there's 2 of them now, are going to have 2 carbons attached. So I've gone from a 3 carbon to a 2 carbon in this intermediate step. Now let's talk about the next couple of steps that are occurring in the mitochondria, or in this case we would say in prokaryotes, it would occur in the cytoplasm and then in the microbe, let's say bacteria, cell membrane. Krebs cycle, otherwise known, it goes by several other names: citric acid cycle, tricarboxylic acid cycle. Okay, this is a series of redox and decarboxylation reactions in which carbon dioxide molecule is removed and potential energy in the form of NADH and FADH2 and several ATP molecules are made. The NADH and the FADH2 will deliver their electrons and hydrogens to the electron transport chain. That's it. Notice how many steps here? Again, no, don't lose sleep. You do not have to memorize all the little steps and all the inversions and everything else. You do get a little bit of ATP made, okay? You get some FADH and NADH made. These will all play a role more in the electron transport chain. But this is part of what occurs. I've taken the 2 molecules of acetyl-CoA. So in each acetyl-CoA, there's 2 carbons, right? So I go once around with 1 carbon, once around with the other carbon. I'm also going to be making, this molecule alone will be carried through, and it will be stripped off of both of its carbons here and here, okay? And what will happen is eventually you have carbon dioxide, NADHs, as I've said, a little bit of ATP. That's the first acetyl-CoA. The second acetyl-CoA follows along, goes through the same cycle, gets broken down, some carbon dioxide made. Now, we're going to start moving into the electron carriers that are required for the electron transport chain. First off, you don't have to memorize everything, get a little bit familiarized, but that's it. Because you're going to have a variety of electron carriers present. The flavin groups, ubiquinone, iron-sulfur complexes, heme groups, and copper, okay? These are either going to take 1 or 2 electrons, okay? Now, the electron transport chain, otherwise known as electron respiratory chains, are coordinated redox reactions that transfer electrons from donors to acceptors while harnessing energy to convert ADP to ATP. Now look, it's this step, the ETC, the electron transport chain, that makes the greatest amount of ATP per molecule of glucose at all. Now, a microbiology test point, the oxidase test can detect one of the electron carriers in the electron transport chain. Some bacteria have this carrier, others do not. If we are talking about the carrier cytochrome C oxidase, the oxidase test is fantastic to help you in diagnosing a particular strain of bacteria, Neisseria gonorrhoeae. If you remember that that's a sexually transmitted disease, the bacteria have basically cytochrome C oxidase, and therefore they're going to be oxidase positive. If you compare that to the results that you just do with the same test with the Salmonella species, they lack the cytochrome C oxidase, and therefore they're going to be oxidase negative. Now, the aerobic respiratory chain uses oxygen as a final electron acceptor. But are there other ones? Yes, and that's where we get into the anaerobic respiratory chains that use other final electron acceptors. In other words, as we've explored bacteria, archaea, in both on surface of the earth, underwater, in sewage treatment plants, underground, you name it, hot springs, etc., we start finding that not all bacteria are the same. Yeah, well, we knew that, but they also have some very unique properties. The anaerobic respiratory chains don't use oxygen, so what is their electron acceptor? It's either nitrate or nitrite, okay, those chemical groups. The location of the electron transport chain, as I mentioned to you before, in eukaryotes, that's the inner membrane of mitochondria. In prokaryotes, it's in the plasma membrane. And that respiratory chains you want to keep in mind are always organized so that the electrons are passing from a higher to a lower energy state. That is how you're harnessing energy. Now, when we get to the ETC, electron transport chain, you're looking inside this inner membrane. Here's the key points. My electron carriers will receive the electrons, and they will tend to take the hydrogens, pump them into the intermembrane space. The electrons are going to be basically added in and doing certain functions for a while. They eventually join up with oxygen and a couple of hydrogens to make water. Okay? Now, as I'm pumping more hydrogens out here, there's a greater amount of hydrogens here in the intermembrane space than in the mitochondrial matrix, separated by a membrane. So what's going to happen there? You've got actually what's called a chemiosmotic gradient. In other words, chemical, but also osmotic, having an effect. You have a gradient, a greater number of hydrogens here, very few hydrogens here. So far, so good. As you go through the ETC with its various basically electron carriers, you're also going to deal with something else here. What are these hydrogens going to do to go from high concentration to low concentration? The interesting thing is that you're going to have this particular large enzyme complex called ATP synthase. In other words, it's synthesizing, making ATP from what? Adenosine diphosphate plus inorganic phosphate. In other words, a phosphate molecule. So as the hydrogens start running through this system, which is embedded in the membrane and has this extension outside of it, this extension here rotates. And as it rotates, it takes ADP and PI and kicks out ATP. And this is as the hydrogen runs down it. It's sort of like almost a hydroelectric dam. As the water goes through, you spin the turbines and you get electricity. In this case, as this structure, these ions go through, they turn the functional components of the enzyme, and it fuses together the ADP and the PI, making new molecules, and many of them, for ATP. Now, as I mentioned before, I mentioned this chemiosmosis, and basically oxidative phosphorylation uses chemiosmosis to recharge ADP to ATP. What chemiosmosis is the movement of ions across a semi-permeable membrane down their electrochemical gradient. And you can see the chart in a bit. The hydrogen ions are pumped into this intermembrane space via electron transport chain. And they have this concentration gradient. A lot on one side, very little on the other. The gradient will run through the enzyme, the ATP synthase, to drive ADP to ATP conversion. Now, this is where we get into what's called the proton motive force. This is the force of protons, that is the hydrogen ions. Because when you strip away the electron, all hydrogen's left is just one little dinky proton. And the force of protons built up on one side of the membrane is that proton motive force. That's going to drive the ATP synthase. So you have a lot of hydrogen here. They start going down through this, and this entire structure rotates. As it rotates, of course, yes, you're going to kick out the hydrogen here. But as it's rotating, it's going to take ADP and PI and make ATP. Okay? Now, aerobic cellular respiration uses oxygen as the final electron acceptor, while anaerobic cellular respiration does not, and I've mentioned that before. You'll want to review the table 8.6 for other final electron chain setups. I want you to keep in mind that ATP production yields for aerobes and anaerobes. When we look at this, theoretically, the process can take one glucose molecule and make 38 ATP. But in anaerobes, generally, you get less ATP molecules that are made per molecule of glucose. So in other words, even though you have a different electron acceptor than oxygen, these structures here that have everything else going, looks almost like what an aerobic cellular respiration does, but they tend to be a little bit less efficient. Here's examples of the final electron acceptors in anaerobic respiratory chains. Nitrogen fixation, that's nitrates. Okay, eventually it reduces this to nitrogen gas. Methanogens, this is in the carbon cycle. This is where they take carbonate and it acts as the final electron acceptor and is reduced to methane. Sulfate reducing, this is in the sulfur cycle. You have reduced sulfate, acts as a final electron acceptor and then is reduced to hydrogen sulfide. Okay? Here is a comparison between aerobic cellular respiration and prokaryotes. Remember how I said the electron transport chain gives you the most ATP molecules per molecule of glucose. Glycolysis, remember you had to do some investment here, and you get ATP made is 4, 2 minus 4 is basically net pathway 2. Intermediate step, you're not making any ATPs. Krebs cycle, you're getting basically 2 per molecule of glucose. Electron transport chain, here's the lion's share, 34 molecules. Okay? And that should help. Now, other catabolic pathways for oxidizing nutrients, there are some other ones, and it's important to kind of be aware of them because, interestingly enough, some diagnostic tests will identify them, and thereby that helps you in your aid of identifying what this particular microbe is. For example, the pentose phosphate pathway uses carbon shuffling to convert pentoses. Now, pent means 5, okay? The pentagon, 5-sided building. You have 5 carbon sugars, okay? And you convert them to trioses, which is basically 2 carbon sugars. You can also convert them also to hexoses, 6 carbon sugars. You can act anabolic and take 5 carbon sugars to make amino acids and nucleotides. So it's because of the pentose phosphate pathway that you can play around and be synthetic in what molecules you need for a particular cellular organism. You will see this in a variety of other cells, Bacillus subtilis, E. coli, and yes, even red blood cells. Now, the Entenier-Dudroff pathway, this is found primarily in gram-negative bacteria that are obligate aerobes. In other words, they have to be present of oxygen. But it's also found in some facultative anaerobes, some gram-positive, and some eukaryotic microbes. It is far less efficient than glycolysis by producing only 1 ATP per molecule instead of 2. Now, when we compare fermentation to aerobic and anaerobic respiration, you can see this table here, which I encourage you to review. You don't have to absolutely memorize, but it should give you some background here. When I look at fermentation, oxygen's not needed. You have a final electron acceptor, but that's going to be an organic molecule. Often it's pyruvic acid. You typically, as a process, get 2 to 3 ATPs gained. Okay. Method of phosphorylation used to make ATP, substrate level only. Okay. Electron transport chain necessary? Nope. Now, aerobic cellular respiration, yes, oxygen, most efficient, get the maximum bang for your buck. You have both substrate level and oxidative phosphorylation, and yes, you have the ETC. Anaerobic, though, oxygen's not needed. You have an inorganic molecule other than oxygen, commonly nitrate, nitrite, carbonate, or sulfate, as the final electron acceptor. Usually you get less than 38 ATPs and more than 2 ATP, though, per glucose. In anaerobic cellular respiration, you have cellular level and oxidative, and you do have an electron transport chain. So let's talk about fermentation. Now, everybody goes, woohoo, booze. No, not just booze. Now, fermentation catabolizes nutrients without using a respiratory chain. The definition of fermentation is the extraction of energy from carbohydrates in absence of respiration. There are different types of fermentation. First one, lactic acid. This is where pyruvic acid is reduced to lactic acid. 2 ATP molecules are made. Homolactic acid fermentation produces only lactic acid. And so that's found in foods. For example, yogurt, the Lactobacillus bulgaricus and the Streptococcus thermophiles are involved with the formation of yogurt. Sauerkraut, Pediococcus and select Lactobacillus species play a role in this. If you're not familiar with these tend to be also techniques to prevent against food spoilage and have been around in some cases for hundreds, if not thousands of years. Remember, we've only had about 100 years of refrigeration. Okay. But if we go from homolactic fermentation, we can go to heterolactic. This is different. We have the production of equal quantities of lactic acid and either ethanol and carbon dioxide, small amount of acetic acid and formic acid, but it's very inefficient. You only have 1 ATP molecule made. It's found in Lactobacillus, Leuconostoc, and Wisselia. Now, with the alcoholic fermentation, yeah, I went there, yeah. This is where pyruvic acid is converted to ethanol and carbon dioxide. It's found in yeast such as Saccharomyces cerevisiae. Sounds like a mouthful, but it's basically brewer's yeast. It's found in plant cells, bacteria, and fungi. You see, think about this. If you were going to bake bread, and I'm going to tell you this real quick story, because when I used to drive through in the evenings in the 80s, through the central part of Worcester, Massachusetts, one of the interesting things is every once in a while I get this sudden smell as I'm driving on the road of baked bread, and I mean lots of it. That's because there was a Wonder Factory, no joke, making bread in the area. Now, here's the deal. When they use the yeast will make ethanol. Now, as also part of the reaction in this yeast causing the bread to rise, what causes it to rise is the formation of carbon dioxide. Carbon dioxide fills all those little holes that you see in the bread. But what happens to the ethanol? Well, the ethanol is evaporated up as the bread is baked. Now, that's part of the problem. You see, the factory got shut down because they didn't want ethanol, that is, the bureaucrats didn't want ethanol to be released into the environment because it's a greenhouse gas. But to this day, we still make bread at home and other places, etc., and carbon dioxide is released. And that's basically the principles. Now, is this the same alcoholic fermentation you make wine, beer, etc.? Yes. But in those cases, we don't mind the CO2 because it may be trapped as you go through the bottling process. But like I said, you can use alcoholic fermentation for a variety of methods, not just beer and wine, but also bread making. Now, another type of fermentation is the mixed acid and butanediol. Pyruvic acid is converted to acids, gases, and butanediol. These acids include lactic, formic, acetic, and succinic acids, as well as gases including carbon dioxide and hydrogen. Now, there's a little story behind this. I want to bring this up to help you and understand how some science can really alter history. There was a scientist at the start of World War I. He was in Britain. Now, Britain, being basically a nation that's surrounded by water, was being beset by German U-boats that would destroy any cargo ships bringing in either materials to make explosives or petroleum or whatever. So the government turned to Chaim Weizmann and said, can you help us here? And he was basically the father of industrial fermentation. What he basically did was set up the capability to do the fermentation process to make large quantities of acetone and butanol and ethanol. And this was using a bacteria called Clostridium acetobutylicium. Now you see these chemicals, acetone and butanol, were eventually used to make cordite, an explosive. And this was so critical for the war effort for Britain that they came to him afterwards and said, okay, you know, you did this for us. Now, I believe this was at the same time, not sort of as an afterthought. And Chaim Weizmann was very much what was called a Zionist, in which he believed in the eventual building of a nation of Israel because he was a Jewish man. And so what he did was he said, well, the British are basically occupying the territory around what they call the Levant, which would be the area around modern-day Israel. He said, I want some sort of honoring of this eventual building of this nation-state of Israel. They said, all right. And they set up what's called the Balfour Declaration, which states that basically the area in the territory of the Levant should be eventually incorporated in some way to the formation of the state of Israel. Chaim Weizmann was eventually also the first president of Israel when it did form in 1948. Now, he had such an influence that he could actually get an empire, the British Empire, to start forging the future for what would be a state of Israel. So that's how impressive his science and how basically it saved Britain by providing the explosives necessary for their munitions to fight a war. Now, as you can see, it all comes down to basically pyruvic acid from glycolysis, whether it's homolactic fermentation, alcoholic fermentation, mixed acid fermentation, or butanediol fermentation. Now, you notice down here where it says cerutin is detected via the Voges-Prosker VP test. This is partly what I'm saying to you. As we get to understand the metabolism, we can use certain biochemical unique steps to identify certain organisms. Stay with us. We're going to continue on this. As we go through this, we come to understand that all cells depend on redox reactions to make ATP. You said that. Now, cells also catabolize lipids, proteins, and nucleic acids. Okay. The term exoenzymes, these are enzymes secreted into the environment, for example, for food source breakdown. Simple example is you see it with fungi. Fungi release a lot of different enzymes outside of their hyphae with the purpose of breaking down the surrounding material, whether that's keratin protein or whether that's in the soil or whatever. And basically they just, by the surface area of the hyphae, absorbing those nutrients as they've been broken down. Now, when we talk about extracellular, we're talking occurring outside of the cell. So the initial catabolism of large molecules can occur. When we have lipid catabolism, the lipase will break down lipids to fatty acids. These are broken down to acetyl-CoA and enter Krebs cycle, and the glycerol will enter glycolysis. What I'm trying to get you to understand here is this. Sometimes some organisms are going to get their nutrients not merely from glucose, but they're going to have to break down lipids, proteins, nucleic acids as part of their capability to obtain the energy necessary to make the ATP. There's also a reversal of that. They will use some of these components to eventually make their own complex molecules in the anabolic synthesis processes. But we'll get into that shortly. One last thought. Each metabolic activity can be detected using selective biochemical tests. When I talk about protein catabolism, proteases and peptidases are enzymes. They break down proteins to smaller peptides and amino acids. The amino acids have to be deaminated. That is, you have to remove the amino group before the molecule can be used in the Krebs cycle. No problem there. Nucleic acid metabolism, these can be broken down depending on the organism. Usually they remove the phosphate group in the sugar. Most organisms will recycle the nucleotides. And so basically you'll have the recycling of the phosphate, nitrogen base, and the sugar. And you can see here, if we start with glucose, we can shift over and not only make some of these substances, but get the substances to help create what you need for energy. Carbohydrates can be broken down to glucose. Other carbohydrates can convert it into glucose. Some of the glucose can be shifted off as an intermediate to make carbohydrates. The same thing here. Some of the intermediates can be used to make amino acids. Some of the intermediates in several of the steps here, both glycolysis and the intermediate step, can be used to synthesize lipids. Also, the Krebs cycle. Now, the other way around is just as equally important. I not only talked with you about carbohydrates being used in the glycolysis cycle. Amino acids, once they've stripped away the amino groups, can be inserted into the glycolysis area. Some of them can actually be inserted realistically in Krebs as well. But lipids can be broken up and they will be inserted into either the intermediate step, some into glycolysis molecules, but a lot of them also into the Krebs cycle. Amino acids here, you strip out the amino acids and you can get them right into the Krebs cycle as well. So let's look at a couple of tests really quickly here. When we talk about exoenzyme profiles, the top set of test tubes are the gelatinase test. This is where an organism is in a liquefied in the gelatin media. Organism A is in that liquefied. You can see how that slopes over. Organism B does not contain gelatinase. All right. And therefore the gelatin stays in sort of a regular level state somewhat. That's where you're going to determine if the exoenzyme is gelatinase here for this particular organism test. Let's go to the middle test. This is an amylase starch hydrolysis determined by iodide. Now keep in mind that iodide, or they might call it Lugol's iodide, IKI, iodide with potassium iodide. Basically, upon contact with starch, it turns black. Now, if it doesn't come in contact with starch, it remains yellow. Now, what you see here is with A makes amylase. It broke down the amylase there, so there was no substrate of amylose present, the starch, so you didn't get a blackened color. Organism B does not make amylase, and therefore when you added the iodide, the surface of the plate turned black. Okay? Now we've got the bottom test is called the spirit blue agar. This is detecting lipolysis. When lipase breaks down lipids, the medium pH will drop. That's because what happens is that if you start breaking down, for example, triglycerides, what are you going to have? You're going to have free fatty acids, which of course have a lower pH, as well as you may have some glycerol. So the agar here is going to precipitate and a dark blue halo forms around organisms. Now, this organism here, A, has lipase positive. Notice the blue sheen around it. Organism B does not have lipase, it's lipase negative. So the pH dropped here, the spirit blue agar had changed color basically into that kind of bluing as the pH of the medium here dropped. Now, we have anabolic reactions. I mentioned that earlier, but let's go through it now for anabolic reactions, biosynthesis. When we talk about biosynthesis, you want to know that the key use of ATP is in many cases biosynthesis. Polysaccharide biosynthesis starts with simple sugars. When I talk about biosynthesis, this is construction of biological molecules. Gluconeogenesis, neo means new. This is where you have the biochemical process of making sugars from non-sugar molecules. Glycogenesis, this is the production of glycogen. This is a branched polymer of glucose molecules. Big long chains of just glucoses attached to one another. Now, bacteria make glycogen by chemically modifying glucose 6-phosphate. This is a glycolysis intermediate. If you remember, I mentioned it earlier. Basically, if you pop some of the intermediates out of the process of glycolysis, you can use them for other molecular needs. But the process to make this glycogen is fueled by ATP. Now, continuing with anabolic reaction biosynthesis, polysaccharide biosynthesis starts with simple sugars. Yeah, we've talked about that. Yep, we talked about that. Yep, we talked about that. Must be a repeat slide. Sorry about that. Let's move on. Here is the glucose. We take some of the molecules, the intermediates, as they're going through glycolysis, shunt them over to make glycogen. And here are these long chains. Each one of these hexagons is a glucose molecule. Now, further down, we take another intermediate and we can now make our peptidoglycan. If you remember peptidoglycan, that's going to be one of the materials that are used in making the cell wall of some of the prokaryotes. Here are the sugars, and here is the peptidyl cross bridges. You'll see this in bacteria, of course. With lipobiosynthesis, this starts with fatty acids and glycerol. The lipids are made in the anabolic process that requires energy. So lipids is basically a process where you have glycerol plus fatty acids. Or another way to say it, though, is triglycerides. That's one glycerol molecule, 3 fatty acid chains added together, you have a triglyceride. You need to note, though, that some bacteria, such as the acid-fast ones, require cell walls enriched with waxy lipids. Example is Mycobacterium tuberculosis. Remember that waxy lipid, the mycolic acid? So this is really strategically important for Mycobacterium tuberculosis to be able to synthesize these unique waxy lipids. And that can be energy intensive, and yet it also slows down their metabolic activity, so their reproduction, remember, is much slower compared to something like E. coli. Okay, because you're spending time making these very special waxy lipids called mycolic acids, etc. But if we take and tap off from glycolysis the glycerol, the intermediate steps we tap off that and make fatty acids, we end up with simple lipids. Here you have it, a triglyceride molecule, 1, 2, 3. Now, there are cells that will use anabolic reactions to make amino acids. Remember that there's a difference between essential amino acids and non-essential. Essential, these amino acids that the cell cannot synthesize. They must be obtained from the environment. So, to grow some of these organisms, they may need their essential amino acids added to their medium so that they can basically grow. Non-essential amino acids, these are amino acids that the cell can synthesize. As such, it's not required in the environment or the culture medium. Now, amination is a process where the cells take intermediate compounds, attach an amino group, so that you can synthesize the non-essential amino acids. Now, intermediate compounds can come from either the Krebs cycle or glycolysis. Then we get into something else here. Remember pyridines and pyrimidines? Oh yeah, that sounds familiar. That's part of nucleic acids, is it not? Yep. A lot of these are not usually made from scratch. So purines, such as the adenine and guanine, pyrimidines, such as uracilthymine and cytosine, you don't get as much de novo synthesis. It requires a lot of energy, which is why. A lot of times what organisms will do is after they've incorporated some other cell, they will start chopping up everything and they will basically recycle the purines and pyrimidines into their own DNA. Here is the pathways I mentioned a minute ago about the intermediates in the process of Krebs cycle, intermediate step, glycolysis to make various types of amino acids. Okay? The purine nucleotides, rarely as it may occur, but it still could occur, you would have the intermediates supplied by the Krebs cycle and glycolysis, the same for the pyrimidine nucleotides. Now, let's talk about amphibolic pathways. They simultaneously will function in catabolism or anabolism. Cells regulate these amphibolic pathways by regulating the enzymes. Now, enzymes can be basically regulated by availability of cofactors, feedback inhibition, allosteric activation inhibition, and phosphorylation, dephosphorylation. Seems like a mouthful, but basically keep in mind that enzymes are not a switch that you flick on and just keep it on all night. Rather, you want to have it as sort of like a volume knob, turning it up when you need it, turning it down when you don't. It's important to keep in mind that the mechanisms that control this, the availability, the function of the enzymes, etc., they are controlling the speed or rate of reactions in the cell. Here's the summary of anabolism and catabolism. Again, you don't have to memorize everything, but I definitely think it's going to help you to go over this comparison and contrast of catabolic pathways, anabolic pathways, and then all of them. Okay? Now, organisms are metabolically diverse. Yeah, you could say that. What does that mean? Well, we've got autotrophs. We've got heterotrophs. In other words, where do you get your lunch? No, it's a little bit more complex than that. Here's one part. How do you fix carbon? See, one way to do it, if you think about just like a plant, is plants fix carbon by taking carbon from carbon dioxide and building up their own organic molecules. So they're autotrophs. They basically can build their own stuff. They feed themselves in essence. Now, heterotrophs can't. They don't have that capability. I can't breathe in air and suddenly start synthesizing my own stuff. I have to get a lot of my organic carbon from a variety of food sources. Okay, proteins, carbohydrates, lipids, etc. Now, that's the point about what we're going to deal with now. Autotrophs versus heterotrophs. Autotrophs are self-feeders. They make their own organic carbon molecules from inorganic start molecules using carbon fixation. Carbon fixation, again, is when they take the carbon in an inorganic form, such as carbon dioxide, and start building complex organic carbon molecules where you have hydrogen that's added on. Okay? Inorganic carbon does not have hydrogens attached. Organic carbon has carbon and hydrogen attached together. Heterotrophs can't do this. They're dependent on outside sources of the organic carbon. In essence, they have to eat other organisms in one sense or another. Now, The initial source of electrons from redox reactions sort lithotropes from organotrophs. Lithotropes. Keep in mind that the initial source of electrons for redox reactions, you need to know. There's different ways. Lithotropes, where you're getting your energy for the process. Well, lithotropes get the reducing power that comes from inorganic sources to fuel reactions. What are some of the inorganic sources? Hydrogen sulfide, hydrogen, or metals? Metals, yeah. And before you think, this is totally crazy, here's one example. Years ago in the, I believe it was 1980s, they were building in, I think it was either North Carolina or South Carolina, a nuclear reactor. Now, they had a hurricane coming in and was going to do all sorts of damage, and they sat there and said, well, what do we do with the reactor shell? Okay, it's not fueled up, but it's this big, heavy, many ton, big piece of steel, sort of like a cup, okay, big container. So they said, well, we'll fill it with water and get out of here. And they filled it with water, figuring that the water would be weight enough to keep the container from going anywhere. When they got back about a week later, they noticed that at the bottom of this huge metal structure was a bunch of pits. I mean, little craters. And they found inside of them bacteria. And the bacteria were living off the metals of this container. And no radioactivity, nothing had been set up yet, nothing had been built, anything else that was radioactive. This was purely organisms that were feeding off the metals for their reducing power, rather than using something from an organic source, etc. Okay? There's also bioleaching, which is another interesting aspect. This is the process of extracting metals from low-grade ores. Lithotropic bacteria do this process. Now, to help you, lithotropic bacteria are very fundamental. You'll take a lot of mines that now produce maybe about, let's say, a couple of grams of gold for maybe out of a ton. But if they spray this particular bacteria and they add water, and after a period of time, they start seeing this strange colored liquid coming out from the pile. That pile is being leached by the bacteria, and the bacteria are releasing the metal ions into that fluid, and as it trickles out, they can collect the fluid and extract out in a much cleaner and quicker way the gold or silver from that fluid. Now, organotrophs, their reducing power is extracted from organic sources for anabolic processes. Now, I'll give you an example. You have to think about the saprobes. For example, fungi. Saprobes are organisms that extract energy from decaying organic matter. Okay? Remember that fungi, when you find like the mushrooms living off the trees and all that, this is a unique situation where the tree trunk may be dead, but what helps break down that organic matter, that tree trunk, is the fungi that are living off of it. They are releasing their enzymes. They're breaking down the cellulose for glucose. They're absorbing that into the fungal hyphae, and it's just incredible. Okay, so we've been talking about microbial metabolism. Let's get to those that use sunlight. Now, how a cell obtains energy to make ATP defines phototrophs and chemotrophs. We've talked about chemotrophs. They use energy and chemical bonds for nutrients to make ATP. You're a chemotroph, I'm a chemotroph. Phototrophs use light for energy to make their ATP. Now, what you see in the bottom there are euglena. These are protozoal, but they're considered more almost on the plant side. Why? Because if you look, they have basically chloroplasts. And I give you a diagram structure of their whole cellular structure. And here is a bunch of them. You would see them be moving around quickly. The flagellum would be whipping them around, etc. And phototrophs are using this light to help make the ATP. Now, there's an unusual group that's called mixotrophs. These are organisms that can shift back and forth from a phototroph to a chemotroph to obtain energy to make ATP. And the example of that is the euglena. The euglena has the capability of not only getting energy from sunlight, but yeah, you could put them in the dark and they could still be doing this as long as you supply them with some chemical bonded, basically organic matter that they can extract the nutrients from and make ATP from. Now, this comes down to this nice little chart. How do we define the terms? Well, let's go for the energy source at the top here. They require organic source of electrons. Do they make organic carbon? Well, let's see. If you have light as your energy source, you will have a photo-organo-autotroph. You could have a photo-organo-heterotroph. The only difference is autotroph, they make their own food. Heterotroph, they have to seek other food. Their ATP comes from photosynthesis. A photolithoautotroph gets it from non-oxygen sources for their electron acceptor, such as hydrogen sulfide, metals, etc. You have a photolithoheterotroph. They've got to get carbon from the outside, but they get some of their terminal electron acceptor from, again, the hydrogen sulfide, that type of thing, metals. When we talk about nutrient breakdown, now, do we make the carbon, do we make organic carbon? Or what? Well, okay. A chemo-organo-autotroph. Okay, I make my own food. I basically make the organic carbon, so I'm doing carbon fixation. Okay. Chemo-organo-heterotroph. I have to get for other sources my energy. Okay. And this may include getting my carbon from other sources. Okay. I can't make organic carbon myself. That's the bottom line here. I can't make organic carbon here either, the chemolitho-heterotroph. The chemolitho-autotroph gets its energy from the lith. Whenever you see that, is basically from, people would think of lith like stone, but you're getting it from non-carbon sources like hydrogen sulfide, like methane, like metals. Okay? Now, here's the key point. Once we start understanding all the different metabolic processes and properties of different bacteria, we can start setting up these nice little biochemical tests. Tests for the metabolic end products, for the intermediates, or for the select enzymes that basically exist in these microbes. And these can be used for the identification of the microbes. There are over 1,000 different tests are available to identify microbes. Amino acid catabolism tests, fermentation tests, methyl red Volks-Prosker test, which is the MRVP. The methyl red is always going to be a pH indicator, so I want you to understand that. So anytime that you're starting to see the pH go down, there's a color change, bingo, if it's got methyl red in it, you're looking for a pH change. The oxidase test, the catalase test, the rapid identification techniques, and the analytical pro-life system, it's the API system. Let me go over a couple of them just to help you here. When we talk about biochemical tests, we have to also start looking at the amino acid catabolism test. This test determines the catabolic activity of the bacteria for selective amino acids. For example, Salmonella versus E. coli on the capacity to remove sulfur from sulfur-containing amino acids. And then they use it to make hydrogen sulfide. So then you would have a chemical reaction with hydrogen sulfide. It might turn black. Remember that you have sulfur in cysteine as an amino acid, okay? Now, fermentation tests, commonly used to identify unknown bacteria by the fermentation of a carbohydrate such as a monosaccharide or disaccharide. They also observe the medium for metabolites. Now, this can include the formation of gas, and this is using a Dunham tube. The metabolites can also be indicated by pH, and therefore you would use a pH indicator. It could be methyl red, but also another go-to that's commonly used is phenol red. The methyl red Vaux-Prosker test, as I mentioned to you before, this is designed to indicate mixed acid fermentation and butanediol fermentation. The bacteria are grown in broth with glucose. They make mixed acids, and therefore the pH indicator will change color. And then you have just the plain VP test, which indicates acetonin, which is an intermediate of butanediol fermentation. Seems like a lot? Let's go over a few points here. Start here. The amino acid catabolism and hydrogen sulfide production. Okay, I'm going to look for something that's going to strip a sulfur from the sulfur-containing amino acid. That's going to lead me to make hydrogen sulfide. Now, hydrogen sulfide combines with iron in a growth media to make a black precipitate. As you can see here, this is a positive test. You see this commonly for a test for Salmonella and for Proteus. Now, in the next one here, and keep note of these little teeny weeny glass tubes here. They're normally filled to the top with the medium, okay? You have a fermentation test here. Now, this is a fermentation where you're looking for the products, and the products are going to cause a drop in the pH. So there you're going to have phenol red. Phenol red at neutral is about red, but when you start dropping the pH, it will turn yellow. Also, when you have fermentation, you will also look for fermentation gases produced as part of the process. Now, these are called the Dunham tubes. Now, normally when you set this up, you've got the liquid in here, you've got the medium, you have the Dunham tube completely filled with the liquid. And so what happens is if there's any gas that's produced, it kind of builds up and starts forming a bubble here. The bubble will be a positive for gas, okay? So organism A fermented lactose. The gas was isolated in the Dunham tube. Organism B didn't do anything, didn't ferment the lactose. We can also use these tests for foodborne illness, Salmonella versus E. coli. They carry out mixed acid fermentation from Enterobacter, which carries out butanediol fermentation. And I mentioned before again, methyl red is also a pH indicator. You have the oxidase test. Now this determines if the bacteria has cytochrome C, which is an electron carrier in the respiratory chains. This test is used to identify Neisseria gonorrhoeae. Okay, yep, the bacteria that cause gonorrhea. Now the catalase test, this is used to identify if bacteria have the enzyme catalase. If you recall, catalase is an enzyme that protects organisms from reactive oxygen species, ROS. Now the test is done by adding hydrogen peroxide to the test specimen. If the catalase is present, the catalase will break down the H2O2 into O2 oxygen, and it'll appear as bubbles present. Oh, and you get oxygen and basically also H2O, water. Okay, so here we have an oxidase and a catalase test. The tests provide the information on oxygen used by the specimen. Okay, on this side here, bacteria that is oxidase positive and generates a purple blue color when smeared onto a dry slide card. Okay, this other organism didn't do anything here, and therefore it's oxidase negative. Here we have catalase positive organism, and it's evidence because what you're seeing is this is liquid. You see the bubbles right here, and that's the bubbling of the hydrogen peroxide present and being broken down to oxygen and water. Now, sounds like a lot of tests, right? The other thing is this. Could you imagine doing like 48 different tests to come to some sort of conclusion of whether it's this bacteria or not? I'm sure you're probably shaking your head going, no way, man. That's why what happened with the onset of speedy diagnostics, the combination of miniaturization of components and parts and test reagents, we started getting this wonderful set of tools called rapid identification techniques. Now, when you have a volume of tests and you're trying to do it rapidly, reliably for the identification of the bacteria, there are now many systems that use a combination of tests that help provide an identification to select bacteria species and even the strains of them. There's several that I want to introduce you to. The API system. This is a rapid, semi-automated tool applied for the identification of foodborne and clinical microbes. Basically, what you have is a strip of tests. They are basically all combined at the same time. You got 20 mini tests. You incubate them for 18 to 24 hours, and the results provide a numerical identification based on positive versus negative results of the tests. And each one of the microbes has a particular numerical identification. We'll get into that in a second. The other one is called an enterotube, and this is using the same approach, but the samples are run through a disposable tube rather than a strip. To help you to see this now, here is the API system. If you look here, you have these little bubbles that contain the reagent and culture. And this test strip contains 1, 2, 3, 4, 5, 6, 7. You can see quite a few tests here. And the small abbreviations here indicate whether they're metabolizing glucose or sorbitol, or whether they are going to do a Vox-Proxer test or urease, etc. Then what happens is you either have it scored as either positive or negative. Okay? So if you have a negative, you have a 2. You have a positive, this is a 1. You have a positive here, this is a 4. So the 3 tests here give you a combined code of 5. And you keep doing this for each of these sets until you come up with, in this case, it would be a 7-digit number. The 7-digit number you can just look up in sort of an index set up by the API system, and basically that will tell you what is the identity of this particular organism, which is Klebsiella pneumoniae. Think about this. You've done about 20 tests in 18 to 24 hours, and you were able to get a solid identification without gene testing. I prefer gene testing, by the way, to anything else, but still, this allows for very rapid diagnosis. Okay? Now, this is used for identification of clinical and food microbiology specimens. This is the enterotube. The difference is you have a tube. Each one of these wells has a particular test for it. Again, the results of the wells will give you a score which provides an ID, the biocode. This one tests for gas and glucose and whether it uses lysine, the amino acid. And so what happens is you go through all of these tests. This one is, do you have production of basically hydrogen sulfide? You notice over there it might be black, that's hydrogen sulfide positive, etc. You continue to go through all of these. And you notice here that none of them came through positive, so that's a 0. These came through positive, that's a 7. These came through positive, that's a 7, etc. All of these tests combined will give you a numerical code, a biocode as they refer to it. I used to do this with my students in a microbiology course. Now the enterotube is focused on the hundreds, if not almost thousands, of different organisms, bacterial organisms you're going to find in the gut. Some are pathogenic, some are not. And what you basically do is you take off the cap here, take off the cap here. You have a culture You dip, there's a metal rod that goes through all the way through here. You dip the metal rod, the tip of it, and pull the entire metal rod all the way through here, which basically inoculates every one of these chambers. You put it into an incubator. Wait 24 hours, you'll get the different types of culture results. And then you have the ID code. You go to the book, open up the ID code for 70773, for example, and you will know what you have. That's how quick it is. And to be honest with you, that's far faster than doing 48 tests. So you imagine 48 Petri dishes, putting them away, and then just examining each one of them, whether you've got growth, what's the color, et cetera, et cetera. And this miniaturizes it. Even basically the footprint or the amount of space that's taken up is far less than 48 plates. Okay, so we've pretty much gone through this entire chapter. You have an exam coming up very shortly, so please note this in your blackboard. You do want to review the visual summary on pages 264, 265, review the chapter overview on page 266, review end of the chapter questions on pages 267, 269. Review the homework in Mastering Microbiology, as well as the quiz for Mastering Microbiology. Peruse the videos in the blackboard for the lecture content. This is going to be very helpful and supportive for you. In our next section, we're going to go into now a new area, the principles of infectious disease and epidemiology, which is chapter 9. Have a wonderful day.